Fonesca and I'm a research scientist in Georgia Tech. So we are part of the systems biology core, and we'll be trying to integrate all the data produced by the experimentalists, and we'll try to come up with a mathematical model that allows us to explain how the infection of malaria occurs. When we construct models, what we are building on hypotheses. We just simulate, so we just see what that hypothesis leads us into, and we just compare the result of the simulation with reality. And if the hypotheses are wrong, then they don't match. If not, if we have a match, then supposedly the hypotheses are correct, and that gives us a different understanding of what reality is, and it gives us a way of then testing other hypotheses. Usually what we get is metabolic data on pathways like glycolysis and Krebs cycle, and what we do is we try to we take information from databases on the enzyme kinetics, so how fast each reaction occurs, and we try to build a model of the whole pathway, and in that way we can predict what happens is if an enzyme is not there, or if the amount of enzyme is reduced, even if not totally, what will happen. We have used these to change the Lactococcus lactis bacterium, for example, that is used to ferment yogurt. And with these techniques, we have produced bacteria that are able to naturally sweeten the yogurt by releasing glucose. It doesn't consume all the glucose that is present in the milk, and so we are trying to use these techniques also to construct Lactococcus lactis strains that are able to produce important factors for human health, and in that way we can produce yogurts that naturally come with these things, so that a person doesn't need to take them pharmaceutically, and we can just have them as we are eating a yogurt. So right now I'm a modeler, specifically on metabolic models, and as an undergrad I studied biochemistry, and I did my Ph.D. on biochemistry also, where I studied the metabolism of brain cells by NMR. This technique allows us to get time series data on different metabolites within the same pathway. And so this allows us to get very dense time series data, very rich in metabolites, which then became very important to modelers. And so from a collaboration with a group here in Georgia Tech, I then, instead of just doing the experiments, I started also collaborating with them, analyzing the data, and I liked it, the more dry side, and so I just decided to change my work and do some more modeling research. So this project, I think the biggest key here is that we are all aiming at malaria, but with a whole bunch of different omic techniques: proteomics, genomics, transcriptomics, metabolomics, lipidomics, and of course all these omics data, they don't make sense if we don't have a model or a way of explaining what's going on. And I think the biggest advantage in this project is the whole different omic techniques that are going to be used, and then the integrative part of the systems biology. Hopefully we'll be able to make sense of what goes on during a malaria infection, and what is the role of each individual in the, in an epidemic or in ecology of malaria. So all these techniques that are going to be used, these are techniques that are in the forefront of development. Any student that wants to graduate nowadays, they want to have experience with with the latest techniques in proteomics, the latest type of transcriptomics, and so this project is again a great advantage for them as a training opportunity. Usually, either things start with an idea from the experimentalists. They started studying something, they produced the data, and they come to us with data and a question. And so in that way, our task is more to try to answer that question from their own data. Or often from our own research, we developed questions which then require data to be answered. And so we go to them and we ask for them to study a particular aspect, and if they can, they supply us with data. And then as we get data and as we talk with experimentalists about the system that they are studying, we get new ideas of why the model might not be correct, or why that type of data doesn't really work into parameterize some part of the model. And so it's a very interactive work where we need to ask all the time why the data or why our understanding of the system is not completely correct. And then we go back and we do our computational things and we make a lot of simulations until we see if we can get simulations to match with what they observe.